In the mid-19th century, Gregor Mendel approached the study of biology with an unprecedented level of mathematical rigor. Instead of viewing inheritance as an unpredictable blending of traits, he documented a rigid statistical system. Mendel tested this system by crossbreeding pea plants that differed in two distinct contrasting traits, such as seed color and seed shape. From these tests, he derived the law of independent assortment. The law dictates that when a hybrid reproduces, individual traits segregate completely independently of one another. If traits segregate independently, the mathematics guarantee a highly specific outcome in a dihybrid cross. The resulting generations will display a precise 9:3 to 3:1 ratio of trait combinations. This animated Punnett square visualizes the exact probability at play. Generating the 16 possible combinations of gametes aggregates naturally into that perfect 9:3 to 3:1 distribution. 50 years later, biologist Thomas Hunt Morgan attempted to apply this mathematical rule to a completely different organism. Morgan bred Drosophila melanogaster, tiny fruit flies, specifically selecting for two contrasting traits, body color and eye color. The progeny of these flies completely failed to produce the expected 9:3 to 3:1 ratio. Instead, the offspring's traits skewed heavily toward the exact combinations present in the parent flies. Why do some gene pairs actively resist independent assortment? The answer to this mathematical failure required a physical remapping of the genome itself. To understand exactly what Morgan's flies were violating, we have to look at the strict statistical logic behind Mendel's initial cross between a plant with dominant traits and one with recessive traits. Looking at this probability tree, an F1 hybrid forming gametes has a strict 50% probability of passing on the dominant R allele and a 50% probability of passing on the recessive R. For the 9:3 to 3:1 ratio to exist, sorting the second pair of alleles must be a completely separate coin toss, unaffected by the first. Multiplying these independent probabilities yields exactly 4 distinct gamete types, RY, 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 and RY, each with a perfect 25% frequency. This framework relies entirely on the assumption that genes are free-floating, disconnected data points within the cell. By 1900, advancements in optical microscopy allowed scientists to finally look past the data and observe the mechanics of cell division. They discovered dense structures inside the nucleus that appeared to double and divide just before a cell split. These colored bodies were named chromosomes. Biologists Walter Sutton and Theodore Baveri observed that these physical chromosomes behaved exactly like Mendel's abstract genetic factors, always occurring in pairs. During anaphase of meiosis I, homologous chromosome pairs physically align at the metaphase plate. Independent assortment works only because different pairs of chromosomes align and segregate independently of one another. The math matches the physical movement. But this introduces a massive structural limitation. A human possesses tens of thousands of distinct genetic traits, but only 23 pairs of chromosomes. Genes simply cannot be free-floating. Hundreds of different genetic traits must be physically anchored to the exact same massive chromosome structure. Returning to Morgan's Drosophila data, the specific traits he selected, yellow body color and white eye color, were located on the exact same X chromosome. During cell division, these traits are not sorted independently. They are trapped on the same moving body, dragged across the cell together. Morgan coined linkage to describe this physical association of genes permanently tethered to the same chromosome. As shown in this modified tree, linkage destroys the independent 50-50 probability. The two traits are forced to travel as a single, undivided package. This physical tethering explains exactly why Morgan saw overwhelmingly high proportions of parental combinations in his flies. Yet a subtle flaw remained in the data. If the genes were perfectly linked, the mixing of non-parental combinations should be exactly 0. But Morgan observed a small fraction still occasionally mixing. Chromosomes are not indestructible solid lines. They possess a mechanical flaw that occasionally snaps the linkage. Morgan termed this process recombination, the generation of non-parental gene combinations caused by chromosomes physically breaking and swapping material. During meiosis, homologous chromosomes cross over. They twist tightly together, physically snapping apart and seamlessly reattaching with exchange segments. Morgan carried out further dihybrid crosses and discovered that the strength of the linkage was not uniform. Different gene pairs broke apart at drastically different rates. This data compares two crosses. The genes for white eyes and yellow bodies showed a recombination rate of only 1.3%, indicating extreme linkage. But the genes for white eyes and miniature wings showed 37.2% recombination, meaning they broke apart easily and frequently. Alfred Sturtevant, a student in Morgan's lab, realized that these recombination frequencies were not random errors. They were a function of physical space. The further apart two genes sit on a chromosome, the higher the physical probability that a break will occur exactly between them. Sturtevant used these failure percentages as literal units of measurement. Gene markers with a 1.3% recombination rate were placed tightly together. The loosely linked genes with higher recombination rates were slid far down the sequence, creating a precise spatial scale. By treating Mendelian failure rates as a ruler, Sturtevant translated statistical data into the world's first physical map of a chromosome. Using recombination frequency to map genetic distance became the standard practice for analyzing biological inheritance. Decades later, genetic maps built on this exact principle served as the starting point for the sequencing of whole genomes, including the Human Genome Project. By measuring exactly how and where Mendel's rigid mathematical laws failed, early geneticists uncovered the physical cartography of life.